Okay, good evening. Let's start. Okay, so it seems like most of you are doing okay. Great, let's start. So I just want to repeat what is expected of you. There are 14 of you at this point. I think you filled only 11 or 10 or 11 of you filled in dates, and most of you have not filled in topics or got my authorization for topics for your assignment. So I want to go again and remind you what is expected. We want you to give us a story. It could be best paper in your field, but I will tend not to authorize this. I really want you to come up with really good stories, as I'm going to tell you one next week. It could be worst papers in your field, famous fraudulent science cases, famous retractions of papers, errata. Basically, we want to have a good story and some lessons from the story. You'll have much better understanding of what I'm expecting from you after next week, but I really want you to fix a date and come up with a topic and get my authorization for the topic. So time is running out. You are going to start presenting in 3 weeks from now, so please find a topic to discuss. Also, regarding the QM homework, I got 10 responses. That means 4 of you have not submitted the work, and that's not great. So please contact me about your QMs if you have not submitted it. So as I said, please go to the Google Sheet, fill in the date and the topic. Right. Last time we went through Watson and Crick paper, and then we went through Forrester paper. Forrester paper is very important for my own career. So now we'll discuss today a little bit my own work, and that relates to the QM homework. So we said that fluorescence resonant energy transfer is a non-radiationless, it's a radiationless process for transferring energy. It's basically electromagnetic transfer. You have an antenna source, which is the donor, and antenna acceptor that receive that energy without any photons being emitted or absorbed. And the formula that describes the rate of red is given here. It's 1 over the excited state lifetime of the unperturbed donor times 1 over the distance between donor and acceptor to the 6th power. So it's a very strong dependence on distance. We went through the Jablonski diagram describing this situation. And what we'll do today, we'll follow up on the Fred story. So the basic idea is, because of this 1 over r to the 6th power dependence, you have a very strong dependence on distance on the nanometer scale. And that basically provided a ruler to measure distances on macromolecules. And the first paper that showed this is 20 years after Forster theoretical paper coming from Lubert Steyer, who just died recently, and Richard Hagelin. Richard was a student of Lubert and later on went to start up a company that made fluorescent dyes and made a lot of money, sold it to what is nowadays Sigma, and bought a yacht and went all over the world. We report here an experimental study of the dependence of energy transfer on distance. The observed efficiency of energy transfer as a function of distance is in excellent agreement with the 1/r⁶ power dependence proposed by Forster. So this is really taking the idea of Forster 20 years later, building an experimental setup and proving that you can use this mechanism to measure distance. So what did they do? This is, by the way, this paper is published at the end of May 1967, just 1 week before the Six-Day War. They took a peptide, a homo peptide made from the amino acid proline. So it's a poly-L-proline, and they changed the number of proline units in this peptide. So that was a ruler. Basically, proline is a very stiff molecule, and as you chain one proline to another, you get basically a stiff ruler. So that's one of the most stiff known molecules. And basically what they've done, they put a donor and an acceptor dyes on the ends of this peptide for the distances going from 1 to 12 and measure the FRET efficiency. I showed you 3 different methods to measure FRET efficiency on the ensemble level. And here you see the experimental data. Basically, you see as function of distance, there are 12 points here for 12 distances, and you get something that looks really, really close to the 1/r⁶ power dependence. And if you plot it on a log-log scale, you get a straight line. That's basically, the slope that they're getting here is 5.9, really very close to 6, basically proving the Forster theory. However, this was not actually the very first demonstration of FRET in practice. The Weizmann Institute, Edelhoff, Brand, and Wilczek actually published a paper just 2 months before the paper from Stanford. However, the paper from Stanford is much more accepted and known as the very first demonstration of FRET. So here is a conclusion that is drawn by the Weizmann group. Quenching was observed in the series of tryptophan, glycine, tryptophan with ionization of the phenolic group. The efficiency of quenching decreased with increasing molecular size. Quenching is attributed to radiationless energy transfer between the tryptophan and ionized tyrosine residues. So they see here the tail of the full graph that you can see from the Strahan-Hagolan paper. So basically what they have measured is this part of the graph. And they've done it before Strahan-Hagolan, but of course in science, oftentimes the more established group, the better school, better paper that it was published in, and so on, makes the headlines. And until today, the first demonstration of FRET is attributed to Strahan-Hagolan and not to the Weizmann group. All right. So just to summarize, what is FRET? You put 2 molecules, donor and acceptor. They are both dipole emitters, basically. You excite the donor selectively with a green photon, and basically the donor is excited, it transfers energy to the acceptor if the acceptor is really close by, and then you count the number of acceptor photons, and donor photons, and from that you can actually extract distance. In practice, what you do, you go to you put your molecules, let's say polyproline peptide with donor acceptor in a cuvette. You go to a spectrometer or fluorometer basically, and you look at the spectrum of the combined donor acceptor pairs on the peptide. And if the distance is very short, you're mostly going to see acceptor spectrum, very little of donor spectrum. This is a combined spectrum. If the distance is far, you mostly see donor emission and very little acceptor emission, and somewhere in between. The distance where they are equal is called R naught, that 50% energy transfer efficiency. This is when R equals what we call the Forster radius, R naught, which is the parameter. And so this is from 1967 until 1996. FRET gains huge popularity in biochemistry, and people start to measure distances of macromolecules using basically this method. And you can use it for many different things. So it's a ruler. You can actually look at biochemical reactions. If you have basically association reaction, you put on an acceptor on the two participants. You get basically donor and acceptor separate signals. When they associate, you get energy transfer, and you know that the reaction went forward. You can do the opposite. You can have a molecule that's being cleaved by a protease or nuclease. You put on an acceptor on that molecule, the enzyme comes and cuts, and now you lose your FRET signal. So that's basically the idea. And in 1996, my group contributed to this saga, to this story, basically going from ensemble measurement that Schreyer and Hagelin did and all their followers to the level of measuring this on a single molecule. And now let me switch. And so Schreyer and Hagelin experiment is depicted in this simulator. We have a test tube. It's a two-dimensional, not three-dimensional test tube, but it basically demonstrates the concept. You have many molecules, let's say peptide, where donor and acceptor fluctuating. And when you average over a very large number of molecules, the FRET value that you measure is the ensemble level average. So you get one value for the FRET. But if you go to smaller and smaller number of those pairs, you start to see fluctuations, and instead of single value, you get a distribution to the point where you go to 1, and now you see changes in distance as function of time. And that's the main idea. If you are able to do FRET on a single static structural biology to dynamic structure, you can look at basically how the structure of a protein changes in time. So that's basically the idea. Let's go back to the presentation. And the first demonstration was done in a very special microscope called the near-field microscope. There are many reasons. I won't go into the details, but at that point, it was believed that you can measure single molecules only if you have that type of a microscope where light is emanating out of it. This was implemented by taking an optical fiber used for optical communication, coating it with aluminum and leaving the very end open so you have a small aperture. You bring light into the fiber, you collect light with the microscope objective, and basically you raster scan that tip over your sample. And this excites a very small excitation volume. It reduces the background fluorescence, and then you can get actually good signal to noise from single molecules. So that was the very first demonstration. The method to show that you can detect single molecules was developed by Eric Betzig at that time at AT&T Bell Laboratories. Later on in 2013, he got the Nobel Prize. for single molecule spectroscopy and super resolution imaging. This paper, this is a paper he published in 1993. He demonstrated this near field microscope. You have this fiber, the excitation field has this electric field dependence. This is side view, this is top view. He had a glass cover slip coated with molecules, and basically when he raster scan, he could basically detect single molecules and even extract the dipole orientation of these molecules, the orientation of the molecules on the glass surface. That got us to, since we built this microscope about the same time for other purposes, once he published his papers, we put molecules under the microscope, and sure enough, we were able to see single molecules as well. Here you can see some nice photophysics phenomena from single molecules. As you raster scan this fiber tip over the field of view, you see some spots. Those are airy disks, basically showing the points of function of this microscope. And here you see a molecule that was photobleached. It was distracted, and it doesn't emit light anymore, and a few other phenomena. So here you see a nice bright molecule that emits light all the time. Here a molecule that was photodistracted. Here you see a dark state. So you scan, at some point the molecule becomes dark, and then it comes bright again. And here is a noisy molecule. So once we proved to ourselves that we can see single molecules with this microscope, the next idea was, instead of using polyproline as triangulant, we used DNA, and we connected two dyes, TMR and Texas Red, as donor and acceptor to the two ends of this DNA. And we put those molecules at low concentration on glass cover slips. Again, using the near-field microscope. And here we were able, for the first time, to demonstrate that we can measure single molecule FRET on the single molecule level. And based on this simulator that I've shown you, that really started this whole field of dynamic structural biology, because now you can look at changes in this sense as function of time. So that was the first paper in 96. And here is the evidence that we are seeing indeed the FRET process. So what you see here is a spectrum of those DNA molecules labeled with two dyes, donor and acceptor. The energy transfer efficiency, because the distance is very short, it's 10 bases of DNA. This is much shorter than the R0 for this pair. So initially you start with seeing only the spectrum of the acceptor. Time axis starts here at the back and comes towards us. So initially you see only acceptor spectrum, because you don't see any donor photons. The donor transfer all of its energy to the acceptor. After 3.5 minutes, the acceptor photobleaches, and now you see only emission from donor. That's actually basically evidence that indeed before the photodistraction event, you had 100% FRET between this donor and acceptor, and this is a very nice demonstration of single molecule FRET. So you see here the summary, the spectrum before, the spectrum after, and during the bleaching. And you can see the other cases as well, where donor bleaches before acceptor. So when donor bleaches before acceptor, you're left only with direct excitation of the acceptor. If you raster scan and you image, you have two detectors. One detector looks at the donor photons, the other one at acceptor photons. And you raster scan and you build this false color image, you see individual molecules, and those molecules show red first and green second, because each time we raster scan, we excite the acceptor, the donor transfer energy to acceptor, so you see only red photons, but at some point, acceptor photobleach. And all those cases where you see starting in red and finishing with green means that the acceptor photobleached during the scan. And again, you see this here as photon streams. This is the acceptor photo stream, and then bleaching of the acceptor, and now you see photons from the donor, and eventually donor photo bleaches as well. So why acceptor photo bleaching gives a proof for single molecule FRET? Try, even if you don't know. Guess. Nobody is willing to volunteer an answer? You can open your mic if you want. Okay, overlap between donor and acceptor. Anything else? All right, so let's go back here. Basically, we have this situation. We excite the donor and donor transfer energy to acceptor. If the distance here is short, all the excitation goes here. So we see just red photons. The minute we don't, there is no energy transfer anymore. That means that all the excitation of the donor has to be dissipated by radiative transitions from the excited state to the ground state of the donor. So that really gives us this type of signals, as we've seen here. Basically, we see here acceptor photons, acceptor photobleaches, and now we see the donor photons. So that's a proof for single molecule FET. Later on, we moved from being able to do this type of measurements on surfaces, dry surfaces of glass, to molecules in solution. At this point, the molecules are not immobilized anymore. They're actually freely diffusing in solution. And we had to switch the microscope. Instead of using this fiber tip, we are using a simple, very strong lens where we focus laser light very tightly into the solution. And as molecules traverse the laser beam, they are being excited and emitting photons, but you cannot observe one molecule for a long time. You're basically limited by diffusion. Basically, get into the confocal spot and in and out. And what you get out of this is what we call bursts. You get few photons that you are basically using those avalanche photodiode detectors to count photons. And you use a criteria to define what is a burst. You basically bin your time axis and you look at number of photons per bin, and every now and then you see this burst of photons. That means that the molecule went into the confocal spot and then went out. But you're doing it with two detectors. So whenever you have a molecule in the confocal spot, you can still measure how many photons you have from the red dye, how many photons you have from the green dye. And therefore, for each burst, you can calculate FRET efficiency and therefore distance. So very similar to the experiment that Stryer and Hagelin did, but instead of doing this with peptide, we are doing this with DNA. So here you see 7 bases separation between donor and acceptor, 12 and 19. And here you see a very similar graph to what you have seen from the Stryer and Hagelin paper, except that we don't really see 1 over R to the 6th power. The curve here is, the data points really deviate from this curve quite a bit. So anybody has ideas why this deviation? You can open your mic and answer freely. Let me give you a hint. For the polypeptide, I told you it's a very rigid, stiff molecule. And the dyes connected to the polypeptide were also done through a stiff bond. So the answer is that these linkers that connect the dyes to the DNA are basically carbon chain, and they can rotate and rotationally diffuse. And even the DNA itself is not as stiff as the polyproline peptide. And therefore, we can get fluctuations in the distance, and we average those fluctuations. And in fact, that point, exactly that point, was looked at, again, much more carefully by the group of Bill Eaton and Schuler later on. And here is the first paper. They actually repeated one-to-one the Stroy and Hagoland paper on the polyproline peptide, but now down on the single molecule level. And basically here the conclusion, I mean, the abstract of this paper, the observed mean fret efficiencies agree with those determined from ensemble lifetime measurements, but differ considerably from the values expected from force detected as a rigid rod. So basically, they claim that they repeated Stroy and Hagoland experiment, but they don't get the same result. They are doing a much more accurate measurement compared to Stroy and Hagoland, which did an ensemble average. Here now on a single molecule level, they basically claim that the data of Stroy and Hagoland is maybe, I mean, they don't say this, but who knows, maybe fabricated, maybe a loving eye was massaging this data a little bit, but basically they don't get the same results. So here is the experiment they did. This is polyproline, this is the two dyes, here are the fret histograms, and they get pretty strong deviation from 1 over r to the 6th power. And they come up with a different function that takes into account the flexible linkers and the idea that polyproline itself can undergo isomerization. And each individual bonds between amino acids can be in a cis or in a trans position. And taking that into account, they can fit the data. So they find this dependence rather than 1 over r to the 6th power. And later on, they came back on this. They look at the flexibility of the cis residue in single molecule fret studies of polyproline. We reinvestigate this issue further. In water, 30% of the molecule contain internal cis prolines. So basically, they attribute the main result for deviation from Forster to the fact that the polypeptide is not as stiff as people previously used to think. There are many kinks in this peptide because of the cis-trans transition between the amino acids. We find that the fluctuations of the chromophores attached to long flexible linkers also play an important role. All right, but that doesn't really change the fact that the method, maybe it's not as good for measuring absolute distances unless you do all the calibration, you take into account all the fluctuations and so on. But it really allows you to look at moving parts. And that really started a whole era of using single molecule fret to looking into enzymes, into all kinds of reactions, protein folding, unfolding, aggregates, irregular, unstructured peptides, and so on. So this is an old slide I used to show many years ago, but the movie is not working for some reason. Okay, can you see this now?
Yes.
Can you see the fluctuation of the peptide? Yes or no?
Yes.
Okay.
Yeah.
So this is just MD simulation of alpha helix folding. Here is a very famous enzyme that got a Nobel Prize, F1F0 ATPase, actually a Nobel Prize that went to UCLA in the building I'm sitting in right now. This is basically the molecule that is responsible for making ATP in the membrane of the cell. And these are very early wishful thinking ideas that if we put on an acceptor on those systems, we can see those unique motions. And indeed, after 30 years of work, a lot of that has been done. This is another example of enzyme that process DNA, and we can look at this type of processes. In fact, single molecule fret experiments have touched all aspects of the central dogma of biology. You're all physicists, but you might know that the genetic information resides in DNA. when the cell needs to make proteins, this DNA is being transcribed to messenger RNA by an enzyme called RNA polymerase. When DNA needs to be replicated before cell division, the enzyme DNA polymerase basically copies the DNA. The messenger RNA is being translated from RNA to protein by an enzyme called ribosome. Obviously, all of these reactions won the Nobel Prize at some point, and all of these reactions have been looked at with single molecule threat in great details to shed new light on these reactions. Another Nobel was given to the Technion group for the degradation of proteins. So this is Hershko and Chekhanova in 2009. My group has concentrated on using single molecule threat to study transcription, RNA polymerase. This is the most regulated step in gene expression. So each time the cell needs to make some new proteins for some new function because of signal from the outside, the first step in this and the highest regulated step is making transcription cycle. So you start with the enzyme, the core RNAP. There is a transcription factor called sigma that helps RNAP to find the promoter, the start of a gene on the chromosome. This complex binds to the promoter, separate of the template and non-template strand of that gene. And if you now provide nucleotides, the fuel for this enzyme, it will start to make RNA. So RNA will be copied from the template strand, you see here in red, get elongated, and when the gene is fully completed, it's basically being released, the bubble closes up, the components falls apart, and you are able to go to another cycle of transcription. However, there are lots of dynamical steps that are very crucial. When you start to make this RNA, the enzyme is, think about an old VW chipushit, buggy car starting to go up the hill is very steep, the engine shuts off and the car slides back, try again. Somehow with enough energy fluctuations, it manages to go over the hill, and at that point it's committed and it goes all the way down the hill to finish the reaction. This process is called abortive initiation, and it's basically giving more time for regulation of that transcription process. So we studied this abortive initiation process, and in fact, the cue that you got for homework relates to this abortive initiation. When 7 bases of RNA, it actually hits some kind of a barrier. It needs to overcome this barrier by flipping a part of sigma. That costs energy, and this is really the reason for abortive initiation. But if you are able to cross that part, then basically the enzyme escapes the promoter and moves into elongation. So these are the processes that my group has focused on. And the tool that we've used, I already elucidated to. We are doing single molecule threat in solution. So basically we take a laser beam, focus it very tightly into solution, and we wait for these molecules to come in and out and collect bursts. However, if we have only one color laser, when the molecule comes in, we can see basically the bursts, donor transfer energy to acceptor. We detect photon streams in both donor and acceptor, but here you see this type of bursts, and that we basically, for each burst, we calculate energy transfer efficiency or distance, and we get this type of histograms. So here you see a mixture of 2 DNA molecules, DNA 14 and DNA 7. You can see very clearly that those 2 molecules are distinguished. But there is also a spurious peak where you have donor-only molecules, and you want to get rid of that peak. But no matter what you do in terms of biochemical preparation of your samples, you always have some residual donor-only molecules. And in order to get rid of these, we improved our optical setup. Instead of using one laser as the molecule goes through, and you get a single histogram, you actually alternate 2 lasers very quickly. So you start with the donor cycle, you switch very quickly during the diffusion time. to an acceptor exciting laser. So now you test for the presence of the acceptor. You switch back to donor, you measure FRET efficiency, you switch back to acceptor to test if acceptor is there and so on. You do it very rapidly. And you can define two axes. The first axis is the same axis as before, the FRET efficiency. But from this extra information, you can also define a new axis, which we call stoichiometry. When it's 1, it shows us only the donor-only molecule. When it's 0, it shows us only the acceptor-only molecules. And when it's 1/2, you see the FRET-relevant molecules. So now you can use electronic purification of your heterogeneous sample to look only at subpopulation of interest, such that you can see only distant changes and nothing else. So this method is called alternating laser excitation spectroscopy, or ALEX, and that has become very, very powerful tool. So with this type of approach, we can test motion of enzymes on DNA. We look at the distance between the leading edge of the enzyme and downstream DNA. And here, for example, you can see the transcription reaction and escape from promoter. And as I said, in the paper you got to read for the QM, we are interested in the abortive initiation process. So by the time that we did this experiment, that's around the early 2000s, there are 25 years of a puzzle, big puzzle, which is what exactly the mechanism of abortive initiation. First experiments were done in the 80s by a group here at UCLA, by Jay Gralla, where they have done something called footprinting experiments. I won't go into the details, but those are traditional and ensemble experiments, which were very puzzling. Showed that in initiation, the enzyme is bound to the promoter on the trailing edge, but not on the leading edge. So when you do footprinting in footprinting experiments, you cross-link the enzyme to the DNA, you chop off all the free DNA, and then you purify and you release the DNA from cross-linking, and you analyze the DNA sequences and length. And when you do this, you see that during abortive initiation, the enzyme is not moving on the DNA. If you look at my arm, in elongation, the enzyme basically goes over DNA, but the enzyme footprint is always the same. In initiation, that wasn't the case, and that was very puzzling. So when you looked at the protected DNA, you saw many. For 25 years, there was no answer to explain this observation. So people came up with all kinds of different models over those years, and we set up to actually test this and figure out what's going on. So the idea was to basically, here you see the 3 possible mechanisms. There is something called transient excursion. That was one possible model. Enzyme sits at the promoter, makes very brief excursion, and comes back and sits most of the time at the back side of the promoter. So you have those very brief bursts, but most of the time the enzyme sits in the back position. So when you do those ensemble average, time average experiments of footprinting, you see that the back end of the promoter is protected, but the leading edge is smeared. So that's the transient excursion. The other model was inch warming. Basically, the enzyme is like a worm that can stretch and contract during this process. So one end is fixed on the backside of the promoter, and the other end is kind of moving back and forth. So that's an inch warming. And the last model is, it's very similar concept, but it's not the enzyme that basically does this stretching and contraction, it's the DNA. Remember, when the enzyme binds the promoter, it opens a bubble. Now you can pull in on the DNA, and the DNA scrunches in and then released. So those were the 3 models after 25 years of experiments, basically trying to explain Jay Gralla experiments that showed really puzzling results. So now that we have a method to look at dynamic structural changes, we can basically test which of those three models is correct. So again, just to summarize, the inchworming says that the protein undergoes conformational changes. The scrunching says that DNA undergoes conformational changes. And transient excursion, neither DNA nor protein undergo conformational changes. It's just a matter of sitting at the back position most of the time and then making very brief excursion forward. So in order to tell apart those three different models, we did several different experiments. First, we put the donor on the leading edge of the enzyme and downstream DNA. So you can see that in all three models, that distance, as you start to give nucleotides to the enzyme, in all three cases, that distance should get shorter. And that's in a way is also a positive control. We can actually see that the transcription reaction is working. So obviously by these dye positions, we cannot tell apart which of the models is correct, but we can say that the enzyme is active. In the other position, so we put donor on the trailing edge of the enzyme and upstream DNA. And here, basically, in both inchworming and scrunching, we predict that there will be no distance change because the backside of the enzyme is fixed on the promoter. And only transient excursion predicts that here there will be a distance change. And lastly, we put donor and acceptor on DNA, and only one model predict that there will be scrunching of DNA, changing DNA distance. So by doing all these experiments, we can tell apart which of this model is correct. So here's the data. These are the positions of the dye. We start with this complex, so we bind the enzyme to the DNA, to the promoter, we get this FRET signal, and then we add subset of nucleotides that lock the enzyme in position 7. So you can see basically open complex move to position 7, so the distance gets shorter. So basically that's a proof indeed that the enzyme is working and moving forward. And then the test for transient excursion, as I told you, only this test will tell us if this model is correct or not. Do we see a distance change because the whole enzyme is moving forward? So you see the FRET histogram here. We add nucleotides and absolutely no change. So we can already eliminate one out of the three models. So transient excursion is not the right model. We are left between inchworming and scrunching. So here we put the dyes on the DNA and we ask, is that distance get shorter? And indeed, you see here two populations. One is inactive enzyme, the other one is active enzyme. No, sorry, DNA and complex. And now we add nucleotides and you can see that the distance is changed. So this cloud here is moved to the right. And that means that that distance gets shorter. And basically we see very clear evidence for scrunching. This was done on many different dye positions. And basically the conclusion is that the mechanism for abortive initiation is scrunching. The next work is really, I told you that when we hit position 7, there is some kind of an energy barrier. Sigma 17 needs to be part of Sigma 70. Region 3.2 needs to move out of the exit channel of RNA, and that can give rise to changes in the dynamics. So this is another work that basically we looked into this process, and here we measure the kinetics of the reaction. So if we start the enzyme from the start of the gene, we get very fast reaction. But if we march it to position 7 and then start it from there, we actually get counterintuitive result. We see that the reaction is much slower. That doesn't mean make sense. You actually advance the enzyme forward, and now the reaction gets slower. And the reason is for that at that position you have this barrier. And if you add a protein that knows how to clip that part of the RNA that gets stuck, you actually release the barrier and you go back to this fast reaction. So basically, we were able to show that there is an intermediate pausing in initiation, something that was not known before. So again, we were able to basically add this part to the reaction scheme that was not known before. All right. So I just gave you an example from my own group for how and why single molecule FRET is useful. There were all kinds of papers written by many, many groups. So this is basically a summary review that we have written on this method. This is another community effort from 2 years ago to summarize 30 years of this method. Many, as I said, the adoption of this method is really widespread. There are hundreds of groups using this method to solve problems in structural biology, dynamic structural biology. This is taken from Web of Science, citations. There are 4,200 papers just using the method. There are more than 80,000 citations of this method. And the adoption is really very wide. Nowadays, you can buy commercial microscopes that are specialized in single molecule FRET experiments. There are 3 different commercial vendors. There are many future developments, multi-spot setups that allow to do much higher throughput measurements, microfluidic mixing that gives you much faster dynamics measurements. Actually, in live cells, you can introduce labeled molecules into bacteria and measure FRET and enzyme working on genomes inside live bacteria. So we predict that there will be lots of new applications of this method in the future. You can do this type of experiments not only with 2 colors that I've shown you before, don't acceptable, but actually you can do 3 colors and even 4 colors. And here you are already measuring 3 distances simultaneously instead of only 1 distance. So if you have different domains in a protein that move in a coordinated fashion, you can actually measure this coordinated fashion. Another very nice development, instead of relying on molecule diffusing in and out, you get very short bursts, so you don't get enough photons. There are nowadays some kind of traps, electrokinetic traps, that keep a molecule in the center without tethering it to a surface. The molecule still diffuses, but the electrical field feedback always bring it back to the center. Now you can look at the same molecule for a long period of time. And with very fancy pulse laser techniques, you can actually extend the time resolution of the method, and you can go all the way to nanoseconds. This is work from the Ben Schuler group, so you can look at very, very fast structural dynamics changes. And fluctuations, that's actually work from the lab of Eitan Lerner, who used to be my postdoc. He is in the Hebrew. And you can basically look at fluctuations on a fast time scale. There is a community website, all of types of activities, but basically FRET is a method here to stay, as NMR, crystallography, cryo-EM, and so on. So that's what I had to tell you for today. Next week, we are going to focus on fraudulent case in science as an example. And it will take us more than 1 meeting. So we'll start next week. We'll finish 2 weeks. You guys will be presenting. Any questions?
Next week is the Shavuot.
Yeah, so obviously we move it 1 week forward. Anything else?
And the presentation starts, like the student presentations, starts next.
So, okay, let me go back into the calendar. We might need to cram 4 presentations per I'll communicate with you through the announcements on Lomda if there are any changes, okay?
Okay, thank you.
Anything else? All right. Good night, everybody. So we'll see you in 2 weeks.
Good night.
Bye-bye.